Hi everyone, I'm Madison and I'm going to be going over a few aspects discussed in Chapter 9, Sleep and Biological Rhythms, to answer this question. How is slow wave sleep controlled in the brain? First, let's start with the definition of what slow wave sleep is. Slow wave sleep is a phase of sleep in a non-REM sleep phase. This phase has the deepest sleep as well as the highest amplitude of delta activity in the brain. During slow wave sleep, our muscle activity decreases along with our heart rate, and we experience dreamlike imagery. Now, it's kind of confusing to call slow wave sleep a non-REM sleep because REM actually only takes up a very small portion of our sleep stages, while slow wave sleep occurs in stage 1, stage 2, and stage 3, and REM is only stage 4. When we're talking about the delta waves in the brain during slow wave sleep, we're going to look at these right here in green. If you notice, they go up and down very slowly. They're not super sporadic. So when you think of slow wave sleep, you're going to think of these delta slow wave brain waves. Now, when we're talking about sleep, we kind of have to talk about the opposite fact of arousal. There are 5 arousal neurons in the body: acetylcholine, norepinephrine, serotonin, histamine, and orexin. Now, during the slow wave sleep, all these arousal neuron levels decrease. The only one that is slightly different from the rest is orexin. Orexin suppresses drowsiness. The rest of these have to do with arousal in much different ways and just happen to decrease during the slow wave sleep, while orexin actually suppresses drowsiness and is what motivates to keep us awake. And if you notice here, this is kind of the diagram of how orexin neurons can motivate to stay awake. They're a large part of controlling sleep, and when we see an accumulation of adenosine overriding the orexin is when we're able to sleep. We'll get into that a little bit more, but basically the most important thing to remember is that orexin is the arousal neuron that motivates us to stay awake. Now, speaking of arousal, the preoptic area within the hypothalamus is the most involved. The preoptic is within the hypothalamus and is the most involved with sleep than any other region in the brain. Now, the preoptic neurons, which we will call sleep neurons, suppress arousal neurons until sleep occurs. Now, these neurons form inhibitory synaptic connections with the arousal neurons in the brain. The ventrolateral preoptic area has the most sleep neurons in the body, and within them is the group of GABA neurons that again suppress behavioral arousal and encourage sleep. When we think of GABA, we think of inhibiting, so these neurons inhibit arousal. So now we're looking at what the flip-flop circuit is. When we're talking about these preoptic sleep neurons that inhibit arousal and trigger slow wave sleep, these neurons are also inhibited by the arousal neurons that they inhibit, if that makes sense. It's a little bit confusing. Basically, anything that the sleep neuron inhibits can also be inhibited by that. The acetylcholine, the norepinephrine, the serotonin, the histamine, the orexin, sleep neurons can inhibit all those things and also be inhibited by them. This is called mutual inhibition, which creates a flip-flop circuit. In the flip-flop circuit, there are only 2 states. Either the sleep neurons are active or the wakefulness neurons are active. Both types of neurons cannot be active at the same time. You can't be both awake and asleep. So if we look at graphic A here, this is a graphic of the brain being awake. The flip-flop is on. If we look at these 2 arrows here showing mutual inhibition, the arousal neurons are inhibiting the VLPOA. They are not allowing any sleep neurons to be active, and they are inhibiting them. Now, when we flip that mutual inhibition, we come to this graphic on this side, graphic B. Here, the brain is off, the brain is asleep. All the arousal neurons are being inhibited by the sleep neurons. Now, this is kind of a silly graphic, but I wanted to include it because I think it's kind of a great way to talk about the flip-flop mechanism of being on and off. Now, this flip-flop mechanism is quick. That's an advantage of it. It's meant to be quick because it is not very good for our physical safety if it takes us a very, very long time to fall asleep. You either want to be awake or asleep, not somewhere in between. So when we're thinking of a quick flip-flop switch, I kind of want us to think of Patrick Star here. He's awake and then he's asleep. There's no in between, and the change is very, very quick. So anytime you're thinking about the flip-flop from wakefulness neurons being inhibited to switching off to sleep neurons being activated. Think of Patrick Star here, awake and then asleep, awake and then asleep, as quickly as possible. So now when we're leaving slow wave and we're going into the REM stage of sleep, we are experiencing another flip-flop circuit, except this time instead of dealing with sleep neurons and wakeful neurons, we're dealing with REM on neurons and REM off neurons. REM on neurons are located in the sublateral-dorsal nucleus, and the REM off neurons are within the ventrolateral periaqueductal gray matter. So what happens when the excitatory input of REM off neurons decrease as the arousal neuron levels change, REM on neurons are activated, slow wave sleep is going to end, and it switches to REM sleep. So the way that the brain is controlling slow wave sleep is these flip-flop circuits and mutual inhibition. It's able to turn it on and turn it off, transition from being awake to slow wave sleep to REM sleep to being awake again. It's all done with these flip-flop circuits and mutual inhibition. Now, a concept that is talked about a little bit in the book that I wanted to talk about more with information that I found on my own was discussing astrocytes and adenosine. Now, adenosine is a nucleoside neuromodulator, and what happens is that astrocytes stock glycogen, which is what fuels neurons. And the longer that we are awake and the more that we use these neurons, we experience a decrease in glycogen in our brain, which then increases extracellular adenosine. And adenosine inhibits neural activity and promotes sleep. This is talked about a little bit in the textbook, not too much. And the astrocytes specifically are what kind of grazed over. So I wanted to point out this really interesting 2020 study that I found. Twinkling star-shaped brain cells may hold the key to why and how we sleep. So this article is very, very interesting. It discusses how we have thought in the past that astrocytes only supported sleep function, that they didn't do too much in the ability to put us to sleep, wake us up, that they were just kind of a supporting role. The hypothesis of this study was that maybe astrocytes actually mirrored sleep neurons and were more involved than we thought. What this study found is that although astrocytes are not as active as sleep neurons, they do have a serious effect on the need to sleep, especially with the accumulation of adenosine in the cell. And what this study came to the conclusion of is that it's very important to be targeting astrocytes to better help us understand why and how we sleep. So just kind of something to think about when you are studying this is maybe just some critical thinking about how important these astrocytes really are to our ability to sleep.